So Yale requires that I declare my conflict of interest. The university has filed a provisional patent describing an association between one of the genes I'm going to talk about today and decoding performance, and myself and several members of the lab are listed as inventors on this application. So for those of you who I've never met, or have never met me, let me introduce myself. I'm Jeff Gruen. I'm the departments of pediatrics and genetics, and my lab studies the genetics of learning disabilities. We focus on the common ones, beginning with reading and language. And what I'd like to do today is present to you some early results from really our signature project, an ongoing New Haven Lexinome project is what it's called. It's a longitudinal project, and I'd like to give you the fall 2020 update and start with a brief self-explanatory video.
First, we're going to peel off non at the beginning of the word. Now look at this. It's shoal. Shoal. What does it say? Shoal. So I'm going to peel off shoal. Shoal. My root is so. My whole word is not social. What's the whole word? Shoal. Not social.
So these kids, as you can see, are really struggling with reading. And that's a common thing. In fact, worldwide, about 10% of all students struggle with reading in school. These are students in regular classes, in regular schools, and with normal or above normal IQs. Between 60 to 80% of reading performance is due to multiple genetic variants. In other words, reading is a highly heritable trait. Now, the goal of the New Haven Lexinome Project is to find out the extent to which genetic variants correspond with response to intervention through whole genome sequence analysis, behavioral testing, and functional MRI studies. We currently have enrolled about 500 children here in New Haven. We are and have been doing serial cognitive batteries that assess reading, language, executive function from grades 1 all the way in the same kids through grade 5. And for each child, they've given us a saliva sample from which we've extracted DNA and done 30x, we completed 30x whole genome sequencing and the beginning of those analyses. So what I'd like to present to you is our initial genome-wide association study, or GWAS, predicated on a decoding composite score. Decoding is the way we initially learned to read, and our specific score that we're using are from mainstream tests of reading. They're the Woodcock-Johnson subtest, the Tower subtest, and a test of actually reading comprehension or accuracy. All our studies I'm about to present to you today are adjusted for age, for race, for ethnicity, for sex, and for socioeconomic status. So here are our initial results, presented in a way in which typically GWASs are presented, with each one of these dots representing a single nucleotide polymorphism. There are about 1 million of them. With chromosome 1 here, those SNPs that map to chromosome 1 here on the left, those on chromosome 22 here on the right. And our results here showing a single peak of association here on chromosome 19. And what you can see here is that even one of our SNPs, even though there's a small study, has exceeded the genome-wide significance, or the Bonferroni threshold, at 10 to the -8th. So the first question we have to ask are, could these results be due to error from unaccounted population stratification? And so to address that, we initially looked at the QQ plot, looking at expected versus the observed frequency of SNPs. And you can see they follow the line beautifully, except for this one little peak, which happens to coincide beautifully with our peak on chromosome 19. We also computed the genomic inflation factor. In fact, there is no genomic inflation. It's 1.04, which is more than reasonable. We also began to look at our subpopulations themselves. Our families do a self-report and suggest that they're of African, European, and Hispanic ancestries. So we went to the 1,000 Genome Project, and we got the superpopulations for the Europeans, the African superpopulation, and the Hispanic superpopulation, and we compared the genotypes from our New Haven Lexinome Project kids to those, and you can see as they line up really beautifully, so that now we control for population stratification with 10 principal components, is what we did in the data that I've shown you. So could these results be due to error? Well, I reviewed the QQ plots, we checked for genomic inflation factor, we mapped our New Haven Lexinome Project subjects onto the 1,000 genome populations, and we corrected with 10 PCs. We controlled for socioeconomic status. In data that I don't have time to show you today, we looked at the allele frequencies for our hits in the same subpopulations, and they're all the same. We also looked at decoding performance in each of our subpopulations and their equivalent. And then we did the ultimate. We replicated our results in the genes reading or GRAD study, which has about 1,350 African-American kids and Hispanic American kids, all with similar phenotyping. So we're confident that this is not due to error, and this is a really good hit. So what genes actually correspond with this peak here in chromosome 19? Well, we can look at that under higher magnification. Here's the chromosome itself. Here's this little box where our hit is. And if we look there, what we can see here is that we're looking at a region of about 200,000 bases in length. These are their SNPs down here. These are the markers. And these in the middle here are the genes that they hit. And you can see that almost all 31 of these SNPs hit this gene that was anonymous to us to begin with, called KIAA0355. And in fact, our peak SNP that hit 10 to the -8 is smack in the middle of that. So that's a pretty good indication that that's a gene, but that's not the only indication. The next thing we did was we went to GenoSkyline to look for epigenetic regulatory regions. And in fact, what we found is that over our gene, KIAA0355, in fact, there are very strong epigenetic regulatory regions, and they're active in those areas that are important for language and for reading: the inferior temporal lobe, the angular gyrus, and the dorsolateral prefrontal cortex. The other genes did not. We also looked at gene expression in the GTEx database. And what we saw is that we have high expression in the brain, but significantly and interestingly in the cerebellum, which was kind of a surprise. But it does lend credence to the old cerebellar theory of reading disability, which was developed in the 1990s and proposed by Nicholson and Fawcett because of their observation and others that reading disability is associated with deficits in cerebellar-related functions, such as dystonia, time estimation, skill automatization, obviously I have a problem with that, and balance. And they proposed a cortical cerebellar circuit that probably has an important role in reading. So I showed you a little video of our class. This is one of our intervention classes at Strong Elementary School, kids who are in the 3rd grade and really struggling, although it's the end of the year and they're doing very, very nicely. And what I tried to show you is that we've extracted DNA from them and their entire class of over 500 kids. And we done a genome-wide association study. But now it's time to bring the data back to our study, back to our kids, and see what the implications are. So as I mentioned, this is a longitudinal study. The Lexinome Project is a longitudinal study beginning in grade 1 and following the same group of children in over 20 different elementary schools, all the way through grade 5. And we've done these serial assessments of reading performance represented here by these 4 graphs. This one, letter word ID and word attack, are decoding, your ability to really decode sounds and words from letters, which we do, and we particularly do it when we're beginning to learn to read. Over here is something called comprehension, our ability to comprehend what we're reading, and reading word fluency, how quickly we can actually read through a paragraph. And what we see here is that virtually at every time we assess these, at the first beginning and end of grade 1, all the way through the beginning of grade 5, children with the risk allele for our gene, KIAA0355, which we identified with our GWAS, perform significantly below their colleagues that don't have that risk allele. And they perform that way all the way from grade 1 all the way through grade 5 and don't really ever catch up. In particular, if we look at comprehension and we look at decoding, they really never catch up. And I should mention, this is clinically significant as well. So this is really pretty supportive of what people have proposed as the language reading continuum. And for those of you who have children, know this pretty well in that at young ages, we really focus, this is really ages 1 to 3, we really focus on oral language development. We do this really through phonology and practicing and repetitive ways in which we talk to our children. As they get a little older, they develop a language, begin to put words together, and then they begin to process phonological sounds. And we begin to teach them, as certainly when they hit kindergarten in the United States, knowledge about letters and sounds associated with those letters is what's something we call print knowledge. This is what we're doing in our age group, that we're testing in the New Haven Lexinome Project. This is the group that's learning to read. And here, what's really, really stressed and what we really tested was phonological awareness, also known as decoding, phonics and fluency, as well as language development. And this is where our kids are really falling down and where KIA 355 really seems to be really, really important. Of course, as kids get older, then more advanced and more sophisticated levels of reading and then progressing to creative writing as well. What's interesting is that these developmental time points correlate really nicely with development of the brain, and specifically in those circuits in the brain that are important for the development of language and reading. These are depicted here stylistically from neonate to 2-year-old to an adult. And what you can see here is that not only does well, that's not depicted particularly well here, but the brain, of course, increases in size. But more importantly is that these circuits form and they remodel between the ages, particularly of the neonate into the late adolescence. And those coincide beautifully with these developmental stages. It also gives us the opportunity in our lab, and we do this, to do imaging genetic studies on these circuits. So let me talk a little bit about ongoing computational projects in our lab for people who may be interested. In the Lexinome Project, we're just starting to unravel our data. We're just starting to do the sequence analysis. We have all 500 whole genomes of these kids, and we haven't yet performed a rare variant analysis, so we're going to be starting to do that in the fall of this year. And of course, we're going to connect that up with different behavioral phenotypes, in this case, an assessment of passage comprehension. The other thing that we're going to need to do in our future, we're starting here in the fall in the Lexinome, is to begin to look at copy number variants, polygenic score, GWAS and candidate gene analysis or additional GWASs, and different phenotype composites. That's going to be very, very important, particularly in indexing these to larger biobanks, in particular the UK Biobank. We also do pretty traditional linkage analysis, contingent on learning disabilities from families that we've accumulated over the last 30 to 35 years. Here's from a paper recently published by one of our postdocs in the lab, of a large, complicated family. All the dark circles or the filled in squares and circles were the individuals who had a complex verbal trait disorder. But if we focus here in the middle two pedigrees, what we can see here is that phenotype, in this case, a verbal trait disorder, is really segregating in 3 different chromosomes on this complicated family, spelling on chromosome 1, and something called non-word repetition on chromosome 13. So again, back to our families, sequence analysis. And then what we do is we can bring that back to MRI studies in our same group of children. We've done this before. This we published 2 years ago on connectivity analysis using a regulatory region from a well-known language and reading gene. And what we show is that having a particular allele of that regulatory element, which actually does change expression, correlates beautifully with the strength of the connectivity between these 2 nodes on a well-known reading language circuit. Lastly is that we began to model the variants that we're identifying in human embryonic stem cells that have been differentiated into human neurons and brain organoids. This is some of our most recent data. This is from Adriana Cherskov in the United Sestan's lab, where she actually took KIA 355, and she's staining here in green cells from one of her organoid cultures. And so again, the importance is to bring that back to, and this information back to the classroom. So because we have organoids and human embryonic stem cell neurons, that brings us into single cell RNA sequencing analysis, which hopefully will begin this winter. I should also tell you, we do also have projects outside of our Lexinome studies. This one is on language evolution, in which we regress linguistic features onto population genotypes. And what I mean by that is a sampled data set that we published just a few months ago in Proceedings of the Royal Society of Biology. And here what we're looking at is different populations, actually 50 different populations. And on this bubble map, we're actually mapping three things: location in the world map by longitude and latitude, the size of the bubble, which correlates with the frequency of a transcriptional regulatory region that's important to reading and language, and the frequency of consonants in that particular language. So Igbo has a frequency of about 23 or 24 consonants in their language, whereas Japanese is a little closer to 10. And so we've done that with a lot of different populations around the world. Here you can see Eastern populations, here more Western populations. And then what we're able to do is to actually regress these and show that this particular regulatory feature and the frequency of this particular regulatory feature correlates beautifully with a very nice p-value with the numbers of consonants in specific languages around the world. And we published that actually in PNAS 2 years ago. So with that, I'll stop. I'm happy to take questions. These are all the wonderful people that do all this work. Big shout out to Andrew Adams, who's doing the very nice work in KIAO 355. These are our funders, including the NIH, the CLDRC, and of course the Mann family. With that, I'll stop, and I'm happy to take